Hello friends, welcome to BMH Learning. This video is about Anfinsen's dogma. Anfinsen's dogma, also called as the thermodynamic hypothesis, is a postulate in molecular biology. The dogma states that, at least for smaller globular proteins, the native structure is determined only by the protein's amino acid sequence. This amounts to say that at the environmental conditions, that is, temperature, solvent concentration, and composition, etc., at which folding occurs, the native structure is a unique, stable, and kinetically accessible minimum of the free energy. The three conditions. Uniqueness requires that the sequence does not have any other configuration with a comparable free energy. Hence, the free energy minimum must be unchallenged. Stability. Small changes in the surrounding environment cannot give rise to changes in the minimum configuration. Kinetical accessibility means that the path in the free energy surface from the unfolded to the folded state must be reasonably smooth, or in other words, that the folding of the chain must not involve highly complex changes in the shape. How the protein reaches this structure is the subject of the field of protein folding that has a related dogma called Levinthal's paradox. Levinthal's paradox states that the number of possible conformations available to a given protein is astronomically large. Effectively, this makes computational prediction of protein structure by evaluating all possible conformations unfeasible even for relatively small proteins. Also, some proteins need assistance of another protein called a chaperone protein to fold properly. It has been suggested that this disapproves Anfinsen's dogma. This was all. Thanks for watching.